hydrogen bonding. In this video, we will define hydrogen bonding and identify which molecules have hydrogen bonding. Then we'll give an example that are important in nature of hydrogen bonding. This is the strongest of the intermolecular forces. Hydrogen bonding is similar to dipole-dipole forces in that it is caused by a permanent unequal sharing of electrons, or a polar bond. However, it is special. NO and F, or nitrogen, oxygen, and fluorine, are so electronegative that the bond that they form when bonded to a hydrogen is so polar that we get to give it a special bonding type, called hydrogen bonding. It is very important to note, however, that this is still an intermolecular force, so the force that is occurring is between two different molecules. These, just like dipole-dipole forces, are being constantly broken and reformed. As when I drew dipole-dipole forces, I drew a static picture, but you are imagining it rotating around and interacting with all of the molecules near to it. The red dots that I've drawn in several places are your hydrogen bonds. They are between two different molecules, not within the molecule itself. If it was within the molecule itself, it would be an intramolecular force or a bond. There's one other thing that we need to talk about with hydrogen bonding, and that is the concept of a hydrogen bond donor and acceptor. The atom that is attached to the hydrogen, that is adding to the hydrogen bond, is the hydrogen bond donor. So in this picture that I have drawn, the oxygen, which is attached to the hydrogen that is donating it to the bond, is the H-bond donor. Meanwhile, the atom with the partial negative charge that the hydrogen bond, or that the hydrogen is bonding to in its hydrogen bond, is the acceptor. So in this case, the delta negative is on the oxygen that the H-bond acceptor arrow is pointing to. It is accepting the hydrogen and is therefore the H-bond acceptor. Hydrogen bonding is responsible for many, many important aspects of life, especially in biology. In high school, you may have learned how DNA is a double helix and that there are A and T pairs and there are G and C pairs. These pairs of bases are actually held together by hydrogen bonding. In biology textbooks, they look like a little puzzle piece. Here I've drawn them out for you. This is a very important concept that these are held together by these intermolecular forces. Think about what would happen if the DNA strands were bonded covalently. What important biological functions wouldn't work? Pause and think about this before moving on. The reversible yet strong nature of hydrogen bonds allow for DNA and RNA replication. They can be easily and quickly taken apart and put back together by enzymes. This would be much more complicated and slower if they were covalently bonded. Something else to notice is the two types of pairing. You probably know from high school that A's and T's match and C's and G's match. When you look here, you can notice that they do not have the same number of hydrogen bonds. What does that mean about these pairings? Which one is stronger? The A and T pairing, which is in green and pink, or the C and G pairing, which is in red and blue? Think for a moment and pause and continue when you're ready. Because the A's and T's have 2 hydrogen bonds, but the C's and G's have 3 hydrogen bonds, this means that the C-G pairing is much stronger. And this actually comes into play in molecular biology. We're not going to go into all of that, but keep this in mind for when you get to your later biology classes if you're headed in that direction. Hydrogen bonding also plays important roles in the secondary, tertiary, and quaternary structures of proteins. Since this isn't a bio class, we are not going to be going into the structures of proteins, but just know that they are made up of very long strings of amino acids. I've drawn one generic amino acid here for you. The R group can be a variety of different things. Hydrogen bonding occurs in a variety of areas and can connect one piece of the molecule to another. If you look carefully at this protein structure, you can see little yellow dotted lines. That is where the hydrogen bonding is. This starts to create the complex structure that is required for the molecule, the protein, to do its function. Without these hydrogen bonds, the molecule would not be able to form these complex structures. Let's do one more example in nature. The cellulose that makes up plants is formed from many glucose molecules combining into long chains. These chains, however, would certainly not be enough to support even a small tree all on its own, let alone something like the large redwood forest that we have pictured here. They need support. One of the ways that they get this is from the fact that the OH groups that are in each chain of the sugar molecule, or the cellulose, can associate with the OH groups on the other chains. This forms long fibrils that are extremely strong. There's certainly other ways that trees use to be stronger as well, but this is one of the main ways that it works and has to do with hydrogen bonding. In review, hydrogen bonding is an intermolecular force that occurs whenever you have an H bonded to an N, O, or an F. However, it is still an intermolecular force, and it occurs between two different molecules, not between the NO or F and the H that we were talking about above. It is similar but stronger than a dipole-dipole force, and it plays important roles in all aspects of biology, including DNA, protein, and complex carbohydrate structures.